Well, without further ado, tonight we are lucky to have Mario Lupino. He is a wild bee ecologist and a conservationist at Washington State University. He received his PhD in entomology at WSU. His work has focused on wild bee parasites of eastern Washington and western Idaho, and he is currently studying conservation plantings adjacent to apple orchards in the Columbia River Basin. Please welcome Mario.
Thank you for having me. I am very freshly minted. I just got my degree like 3 weeks ago. So thank you for this is my first big presentation, so I'm excited. Today I'll talk to you about bumblebee parasites and conservation in the Pacific Northwest, as well as just kind of some general bee ecology and the work that's being done to conserve them, not only in the east part of the state, but also in the central part of the state. Okay, so we are going to go through why bees are important first and foremost. They pollinate about 70% of the foods that we eat the most of, along with 87% of the flowering species that keep our planet functioning normally. Unfortunately, most of the crops pollinated by bees are at risk for decrease in production due to pollinator decline. You may be asking, what drives pollinator decline? There are four main factors. Habitat loss, largely driven by human activities, affects bees' access to food, nesting sites, and/or overwintering sites. These habitat requirements are distinct from one another, and a large field of flowers does not necessarily equate to a large bee population. Bees face threats from endo or internal and ecto or external parasites, both micro and macroscopic. They may steal resources, spread disease, or attack the bees themselves. However, some of these organisms can be symbiotic and may help their hosts. Bees exposed to pesticides can die if exposure is high enough, but often face sublethal doses that affect memory, learning, flight, flower handling, nesting activities, and overall health. And actually, the images of those bumblebee colonies, there are some studies done in the late teens that show that if bumblebee colonies are exposed to imidacloprid, they stop making those wax canopies over their nests, and it really hinders their thermoregulation. So they become more sporadic in their movements, and they stop heating themselves and cooling themselves, and it really disrupts things. So the bumblebee colony pictures were really cool. Okay. So lastly, pathogens may negatively harm wild and managed bee species. Pathogens make bees sick, possibly reducing productivity and lifespan. My research has primarily focused on habitat loss and ectoparasites in wild bees of central and eastern Washington, as well as parts of western Idaho. Today, we'll just focus on these topics, and first we'll start with how they impact bumblebees exclusively. With more than 600 species of native bee in the Pacific Northwest, bumblebees are amongst the largest bodies and easiest to spot, as I'm sure all of you know. They're cousins of honeybees, often with larger, fuzzier bodies, and can fly in cooler, wetter conditions. They help pollinate important crops in our region like apples and pears. There are roughly 30 species in Washington, Oregon, and Idaho, with 49 species in the United States. Bumblebees, like many other species of bee, are under threat from pesticides, habitat loss, pathogens, and parasites. Pathogens and parasites can live on or inside of their hosts. Pathogens often cause illnesses that affect bees' behavior and reduce their lifespans, but parasitic mites can help or hinder their hosts by eating other parasites or by stealing resources. For the next section, we're just going to talk exclusively about external mites and pathogens and what you can do to help keep bumblebees safe. Before we can begin helping bumblebees, we first must understand how they and their parasites exist within their environment. Foundress queens, or queens that have mated but have not yet started a colony of their own, overwinter slightly below ground, buried in leaf litter, or in any cavities they may find, along with any parasites and pathogens she may be carrying. In the early spring, the foundress queens gather nectar and pollen and create wax cells to lay their eggs in all by themselves. Many species nest underground, typically in old rodent dens, but other species are known to nest above ground in pre-existing cavities. Once a nest is founded, mites and parasites disperse into the nest and begin their own lives. The queen cares for her developing young, called brood, warms the nest, and gathers resources until her worker caste daughters are old enough to take over these tasks. All the while, the parasites and pathogen populations also grow and then disperse between nestmates, between colonies, and between species. Flowers serve as transmission points, a location numerous bees will visit and immerse themselves in, allowing mites to move between hosts readily. When more flowers are available, the less bees share flowers, and the less likely they are to get sick. Once the workforce is of sufficient size, the queen remains inside the nest to lay eggs for the remainder of her life. The colony reaches its peak size in late summer, with mites and pathogens reaching their maximum number shortly thereafter. New queens are reared from eggs which receive special care from workers, and drones, or males, are reared from unfertilized eggs the queen lays before she dies. The reproductive cast leaves the nest to mate once reaching maturity in the early fall. The colony's old queen, the workers, and the drones that were not successful in mating all die in the fall, and the new foundress queens overwinter individually along with the mites and infectious organisms that are adept at finding them. During mating, any mites that were on males will transfer themselves to females because only the females live. So mites sometimes will preferentially look for males in the nest so that they're more likely to find an overwintering queen. Mites can often be seen with the naked eye and can be found on bees' abdomens, between their thorax and abdomen, and even along their wings. We'll go over the effects that mites can have on their hosts in the next few slides. There are a few different types of mites in the Pacific Northwest, and they each interact with their hosts differently. Mites in the genera Anetus and Emperipes are often beneficial mutualists of bumblebees in this region. That means they coexist peacefully with their hosts, feeding on parasitic fungi, other microorganisms, and bee refuse. Some fungi in bumblebee nests like to consume pollen, and these mites eat that fungi, preventing mass outbreaks that would destroy pollen resources. Other microorganisms can also feed on pollen or the bees themselves, and these mites can help keep those populations in check too. But it's important to note that this is dependent on the mites' population levels and their life stages, where sometimes the mite, if it's a certain age, will act beneficially, and if it's another age, will also attack the resources that bees need. So it's a very touch and go kind of relationship. These mites are considered helpful, and these photos were taken by myself and other researchers at WSU to catalog the diversity of mites in this region. Many species here have not yet been identified, and so any of the mites that you see here, they only go to the general level because species level is almost impossible with these things. So Cerophagus and Proctor laylaps are genera that live alongside bumblebees in their nests. These taxa likely only use bees to disperse through their environment and coexist in their host nests without helping or harming them. Mites in these genera have not been extensively described, so we don't know much about who they are or what they do. Cousinia, Parasitellus, and Pneumolaylaps are usually considered harmful to bumblebees. These mites largely act as kleptoparasites that steal resources from the nest. This behavior can expose food stores and developing larvae to parasitic fungi. This means that not only do these mites harm bees directly, they increase harm by leaving the colony's food and young exposed to further parasitism. Parasites may even feed on bee larvae directly and can transmit pathogens. Many mites even have a special attachment organs, like those noted on Cousinia, for holding onto their host or each other during dispersal activities. However, mites aren't the only parasites bumblebees interact with. Some parasites kill their hosts directly. There are 3 species of cuckoo bumblebees in this region that do just that. These are species of bumblebees which parasitize other bumblebees. Cuckoo bumblebees can enter a nest, kill the resident queen, and force her workers to raise her parasitic progeny. Other organisms are parasitoids, or species which kill their hosts in the process of using them for resources. Flies like the Canopidae and Floridae flies in the Canopidae and Floridae families, and Braconidae and Eulophidae wasps are parasitoids that insert their eggs into foraging bumblebees. The resulting larvae modify their host's brain, forcing the bee to dig its own grave before it's eaten alive from the inside. The parasitoid overwinters in that stage, and they cut a little hole in the exoskeleton of the bee's corpse before they pupate so that they can emerge successfully as an adult in the spring. Additionally, parasitic fungi and bacteria can cause lifelong disease and can transmit between wild and managed bee populations. These organisms also change bee behavior. They can cause confusion, difficulty learning, and reduce lifespan. So the information I just talked about is available in my extension publication, which delves much more in depth into the topics we've just discussed. If you're curious about these topics, Bumblebee Parasites and Conservation in the Pacific Northwest is available for free through WSU's extension website. You can also email me directly if you have any additional questions, or if you find any bees that have gross parasites on them. So if you have those Strepsiptera pictures, I would love to see them. And I'll pause here for questions because we're going to cover like three different uh, topics that all tie together, but I figured I would pause just for questions now if there are any. Does anybody have any questions or should I move on? Let me check the chat.
Well,
We're good.
actually, Sarah Rogers says, I'm so glad I don't have any parasites that do that to me. Well, yeah, but we do have parasites in our guts and whatnot,
Yeah,
so we're not unique.
you're not.
Bumblebees are not unique. And Will is typing away. I don't see any other. Where does Nosmia fit into this? That's a
Oh
question
yeah.
in the chat from Will.
Yeah, so that would be a pathogen that affects bees' behavior. So Nosema, I believe it's a fungus that breaks the cells of the digestive systems of bees. And so it reduces their ability to digest food effectively. It causes them to go to the bathroom a lot more. So they are potentially becoming dehydrated. So it changes the way that they interact with flowers. They spend a lot more time on a specific flower as opposed to visiting lots of different flowers, thereby potentially reducing pollination of some things, but increasing pollination of others. My email has a period between the first and last name, but thank you for putting that in. Um, oh, I have it wrong in my presentation, so that's my fault. Okay. Um, Thank you. Okay. Sorry,
And
any other?
yeah, actually, Elise's
Ooh,
children, she asks, why do bees have parasites at all?
that is a really good question. I think that one's kind of difficult to answer because I think everything has parasites. Um,
Mhm,
we
mhm.
represent like an available niche of resources that could be utilized by another organism. So we ourselves are food, and so adult bees are food too, that another small organism could eat.
Yeah, excellent. And then are Mary asks, are bumblebees infected with varroa?
That is a really good question, and we'll kind of touch on this in the next section that I'm going to cover, but the the short answer is they can be. The For the most part they aren't, but varroa mites can use them for dispersal purposes, and there are like photographs of varroa mites on bumblebees from California that I've seen.
And one more question before maybe you move on to the next section, and we'll have time for questions at the end as well. But these are great questions. Ben Lee asks, can bumblebees have strep
Strepsiptera?
diphtheria?
Yeah. So I just went down to Clark College in Vancouver, Washington, and a woman there gave me some Bombas fosnosenskii that had Strepsiptera in them. So I'm really hoping to like rehydrate the bees so I can pin them and then photograph them because I think it's disgusting and also super interesting. So yeah, Strepsiptera are a thing. I didn't cover them in this because I'd never heard of it and I was proved wrong literally the next year. So okay, last
Excellent.
question and then we're good. So
That's it.
I'll move on.
Thank you.
Okay, yeah. Okay, so now we're going to switch over to some more recent work that I've done. Now that I've spent all this time discussing the parasites that bees might have, let's talk about the pollinators of the Palouse and the parasites that I found that they did have. So between 2020 and 2022, I completed a 3-year observational study in the Palouse region of eastern Washington and western Idaho to examine the pollinator parasite communities there. Bees in this region suffer from severe habitat reduction and fragmentation. Less than 10% of natural landscapes remain in the Palouse. In places where flowers are limited, crops like canola serve as beacons for hungry pollinators. Now, as I've mentioned, studies show that when floral resources are limited and floral usage overlaps, the potential for transmission of parasites is much higher. As such, this study utilized canola to examine wild pollinators and their parasitic communities, with the expectation that large congregation of bees would be more likely to have parasites. Many factors surrounding the parasites, and pollen consumption of bees in this region remain unanswered in the literature. So this study will be submitted for publication later this year to hopefully fill some of those gaps. Namely, this study sought to determine which bees in this region have mites, the mites they carry, and the floral resources bees are utilizing. This study was purely observational and meant to determine a baseline of pollinators and the factors affecting their health in this region. That means I didn't manipulate any aspects of the environment by planting flowers or do something like give bees parasites to determine how quickly they spread between species. All I was interested in was catching bees as they naturally existed in their environment and determining the parasite levels at that level. So in 2020, I sampled 11 geographically distinct locations for a total of 14 collection dates. For those of you familiar with the area, the dark gray blob at the top is Spokane, and that cluster of points kind of in the middle of the screen is Pullman. That's where I was based out of for these studies, or for this study, so that's the majority of my sites. In 2021, I sampled 8 distinct locations for a total of 10 collection periods. And in 2022, I sampled 11 distinct locations for a total of 11 collection periods. Sampling efforts varied slightly between years. In 2022, 2 researchers spent 6 hours each per visit to collect as many bees as they could find. In 2021, researchers caught no more than 30 of Apis mellifera, Bombus, and Andrena, as these were the most common taxa observed in 2020. In 2022, sampling effort was increased to 3 researchers, but only bumblebees exclusively were caught. If fewer than 4 bees were caught in an hour, sampling ceased. For optimal data utilization, these bees were also shared with 2 other research projects. All of that to say, in 2020, we caught all the bees we could find. In 2021, we caught some of the bees that we found. And in 2022, we just looked at bumblebees. It's important to note that the use of wild-caught insects in research does require collection permits in many locations. So I secured permits as necessary for the first 2 years of this project before learning a state permit was also required in 2022. Okay, let's go over what exactly I was doing. I always like to tell people I was frolicking because that's kind of really what I was doing, but we have to make it sound more sciencey. So in the field, researchers walked through flower patches seeking out bees from the desired taxa. They were caught using standard aerial nets and then placed on dry ice for transport. Bees were only caught once they were seen visiting flowers. So if bees were flying around, we weren't interested. We had to make sure that they were in fact pollinating. In the lab, bees were examined for pollen and parasites under a microscope. Any pollen found was removed and grouped into a single lumped sample for each genera at each site. A subset of 16 samples for 2020 and 17 for 2021 were chosen for analysis. Now, like this is very sciencey and I'm not going to get into it. All you need to know is that we looked for a special protein in the pollen, and that special protein is involved in photosynthesis, so it's very distinct to each plant species. So we used reference libraries to assign species information based on the protein information that we got back from this DNA analysis. And that's really all I care to say about it. If you have more questions, obviously ask, but that's the important piece. Okay, so any mites that were found on the bee were removed and slide mounted before being photographed. Mites were then identified to genus using the online Bee Mite ID Lucid key. It's a fantastic key. I wish all keys were like this for taxonomy because you just look at 2 pictures, you look at the characteristic that you're interested in, they're very high quality photos, and you just get to click right through everything that you need to know. So it makes taxonomy much more fun. So during this 3-year study, more than 2,300 bees were inspected for mites, and more than 23 parasites were found. Roughly 7% of bees collected had parasites, and more than 6,500 images were taken of those parasites. 8 genera of mite were found, with Cousinia, shown in figure D on the top, being the most abundant, and Vidia, shown in figure H on the bottom, the least. Additionally, triungulins, larvae from the beetle family Meloidae, were found in small numbers on almost all taxa of bee examined. While the exact effects of mites in this region are unknown, the study will hopefully help direct future researchers towards bees with higher parasitism rates. Next, we'll look at the pollen analysis. Pollen was removed from 506 bees and sent off for DNA analysis, which allowed for the development of a plant-pollinator interaction network spanning 65 unique taxa of plants. This figure shows each genera of bee, their grouped pollen findings on the left, and they're arranged by year, taxa, and site number. The species of flower identified from pollen samples is shown along the top. So while we did clean the nets between sampling efforts, the presence of canola pollen in nearly all samples suggests that there was some cross-contamination between sampling events in both years. Even so, this interaction network demonstrates that even in monocultural settings, bees are visiting diverse flowers. And these flowers or this flower usage does overlap. We'll look at the bee and mite data in a different way in this figure. This alluvial diagram shows the three most common bee taxa and the mite genera collected from them across all samples analyzed from 2020 and 2021. Bees which had mites but were not used for pollen DNA analysis are not represented here, so this isn't a perfect, complete picture of the parasite communities at work. Here we can see the interaction between bee genera on the left and those that had pollen and the mite genera that were on them on the right. The number next to each mite genus is the number of bees with that mite. We can see clearly that Andrena was primarily parasitized by Imperipees, while honeybees had no parasites. Bombus, the largest bees, had the highest number of bees with mites and the most diverse group of mites. So now we'll add another piece of this story. By adding plant interactions, we can see how the three taxa of bee cross paths in their pursuit of pollen and trace the connections between floral communities and parasite communities. With this addition, it's important to note that the number next to each plant species is the number of pooled pollen samples with that pollen type, as compared to the number of individual bees with mites shown in the mite column. So the numbers aren't quite comparable because it's groups and individuals. Now we'll look at the whole network altogether, and I always like to say, just be prepared because this figure is quite elaborate, so it shifts, you have to turn your head. So this shows us the full story, with plant species along the top, bees in the middle, and mites on the bottom. Now we can see the plant species identified from pollen, the bees that had pollen, and the pollen that was collected from them, and the mites that may use those flowers to transmit themselves. Despite frequent overlap in floral communities from all three bee taxa, we can see there's very little overlap in mite communities. Only the triungulin larvae Meloidae was found to utilize two taxa of bee. Again, it's important to note that this diagram only shows the interaction of bees which had both pollen and mites. Mites were present on a much larger subset of bees than is presented here, but we didn't have the exact flower identifications for those to include them in this diagram. Bees were only collected after they were seeing flowers, so it's likely those collected without pollen were probably gathering nectar instead. However, the lack of pollen meant we couldn't include them. So what does this observational study tell us? This study provides a comprehensive collection of native bees in the Palouse, identifying important floral resources and the parasites affecting them. Even in intensely agricultural spaces, wild bees visited a diverse array of flowers. This could mean that bees aren't using all the same flowers like we had previously expected. Most importantly, this study showcases the largest physical and photographic collection of mites on wild bees in the Pacific Northwest. As science continues to become a global enterprise, digitizing collections like this one facilitates cross-country collaboration. More work on pollinators is necessary and should focus on understanding the floral communities utilized by them and their active symbionts, or their parasites. Do we have any questions about bees, mites, or pollen at this
We
point?
do. We've got three real
Okay,
good ones so far. So David Jennings asked, what percentage of bees had parasites? And I'm not sure if you actually covered that in
it
the
was 7.
Okay.
Now, we did the study a little bit uniquely in that we wanted to compare like natural locations to canola, and so those numbers are a little bit different between them, but they weren't done in the same sampling effort. So there were a lot more canola sites than natural sites, so we can't make the direct comparison. But yeah, the average overall is 7%.
Did that seem low to you in advance? Did you have a prediction? Because that seems awfully low to me. When I think about dogs and ticks, it's often that you're going to have more than 7% with, external parasites. So I just was reacting to what I thought was a 7% number that you said.
Yeah, I don't know. I don't think we really have a good baseline number for the number of bees that have parasites on them. I think this is kind of it. There aren't a lot of studies that look at this information. There's been one or two done in California and Oregon. There's been a lot done on the East Coast, but their sample sizes are a lot smaller than mine. And there was one done in Canada that I'm aware of, but it was also like they only looked at maybe 40 bees, so their percentages were usually kind of higher just by the small number of things. Um, I'm also saying that like across all of the taxa that we looked at for bees, it was 7%. But like I mentioned earlier, bumblebees they're bigger and they live in colonies. And so they had a lot more parasites than the, solitary andrina or things that would have been nesting on their own. The fact that bumblebees are social means that there's a lot greater risk for transmission. And so I think if we were to break things down in that way, there would be a different percentage.
I'll go ahead and continue to read a couple more questions. There's a few more that keep popping in, but to continue with David's questions, what was the reason that bees had to have pollen to be included in the analysis? And I apologize if you answered this, I might have missed it.
No, no, no. Yeah. So the only way that we could make that alluvial diagram is that we had all three pieces of information. So unfortunately, we didn't record the flower that we collected bees from. So if they didn't have pollen on them, we weren't able to say definitively the flower that was visited. So that figure inherently shows exactly the flower that was visited based on DNA analysis, the bee that we were able to identify and the mite.
Okay, and I'm going to go ahead and thank you. And I'm going to read
Yeah.
Scott Elderby's question. Do you think the hybridization of honeybees has played a role in the relative abundance of mites in your study? That
No.
hyper-hybridization hasn't made much of a dent in Varroa, though. Does,
Honeybees were the only ones that didn't have any mites. If we go back to this, hold on, let me see if I can do it. If we go back to this figure, you can see honeybees didn't have anything. In the middle there, Apis mellifera had no mites at all. And I think that's largely due to the fact that we clean them. We treat Apis mellifera for mites like crazy. So it kind of makes sense that they're the only ones that didn't have anything on them. They did have triangulins from Meloidae. Meloidae seemed to just kind of be on everything. They were very low values. There weren't a lot of meloids, but they didn't really seem to care which bee they were using to get around. They were just trying to get around. So yeah, I don't think that Apis mellifera, at least in this study, is really playing a role in mite transmission in the way that we might think it is, given the prevalence of varroa mite and tracheal mite and all of their other parasites that are a problem for Apis mellifera.
Thanks. You
okay.
answered my question. My
Cool.
question had to do with the absence of mites on honeybees, not the overabundance.
Oh,
But
I
Oh
said
yeah.
I said abundance. I apologize, Scott. I meant absence. So sorry. I changed the question completely. A couple more.
Yeah.
Let's see. Uh, which of the mite taxa are also present in honeybees' native range? Is it possible that the native bee mites just don't pick them up? That's from Will Peterman.
Yeah, I think, well, so honeybees, Apis mellifera anyway, is native to the Mediterranean. They really like to pollinate olives in like Greece. And that's part of why sorry, my lights just turned off. That's part of why they didn't adapt very well to varroa mites because varroa mites come from Asian honeybees. And so I don't actually know, to answer your question, what their native parasites are. They don't, I would think they didn't seem to have too many because they hadn't developed the same kind of hygienic qualities that Asian honeybees have.
Okay, hopefully, Will, that answered your question.
Yeah.
It, Alex Wickstrom, are there any telltale signs that developed nests would have a non-visible parasite?
That's a really good question. Alex, I would actually love to see the nests that you have.
Hmm.
To be honest, it looks like the nests that you've been getting are pretty healthy. They seem to be kind of like at peak colony size or just before, they're all still pretty clean. Um, I think if you were to look for them, you'd want to check in the pollen stores, that's where I would expect them to be most commonly.
And Mary Anekasoni, I'm saying that wrong. I apologize, Mary. Say again, were you only collecting on canola plants? I'm assuming the answer to that is no.
No,
Okay.
yeah, we collected in canola fields, and then we also, let's see if I can do this quickly. We'll go all the way back. So if you look at this map, you can see the round circle, the circles are canola sites, and then the square blocks are non-canola sites. So we sampled in natural spaces and in canola fields.
Excellent. Okay. The rest are comments at this point. Some good
Okay,
comments. So we'll save the chat for you.
sweet. Okay. So we will move on to, here we go. Okay. Okay. We've talked doom and gloom. So what do we do? Um, we are going to do some things. The good
Mhm.
news is that anyone can help bolster bumblebee populations. Bees need three things: a place to secure food, a place to live, and a place to safely spend the winter. It's important to note that landscapes are mosaics of resources, and so are our neighborhoods. Not every individual garden can meet every need of every bee species. We should all do the best that we can to help pollinators, knowing that the more of us there are working towards the same goal, the better our local mosaic will be. And this picture in the middle is actually a picture from my garden where some bumblebees decided to use a birdhouse that I had. So they'll they'll find spaces if you provide them. Okay, as I've mentioned, bees are under threat from habitat loss, parasites, pesticides, and pathogens. While I've spent most of this presentation focusing on bees and their parasites, personally, I believe the greatest threat to pollinators today is loss of viable habitat. The spaces that pollinators reside in are extremely important for their overall health, as well as their abundance and their diversity. However, humans need places to grow food and live as well. So while our food production has increased dramatically, in parts of Washington, changes in land use have greatly diminished spaces previously occupied by bees over the last century. Fortunately, research has shown that diverse ecosystems provide more consistent pollination services to nearby crops, as bees are able to reside within these spaces and disperse effectively into croplands as needed. This suggests that even though pollinators are at risk for decline, changing our management strategies could help them and help us. Some agri-environmental programs seek to provide these critical habitat requirements for bees, increasing pollinator populations around crops that need it. These flowers are maintained by orchardists to help bees keep bees strong and populations robust in agricultural environments that lack resources. Additionally, these plantings help pollinators move through their environment, colonizing new spaces and establishing populations in landscapes made of monocultures. It's important to note that these effects don't happen overnight. These plantings usually take about 3 years to become established because bees need time to find them, stabilize their populations, and make sure the community itself is stabilized. So if you plant a bunch of flowers this year, it doesn't necessarily mean you're going to have a lot of bees this year. The good news is these plantings are working, and the best thing anyone can do for bees at this point is to provide flowers. Even a 1 meter strip in places not suitable, oh, 1 meter, even a 3 foot strip in places not suitable for crops will make a huge impact. Plantings like these are already appearing throughout the Columbia River Basin in central Washington, and this work has already been featured for its success in a local, what's the word, publication called The Good Fruit Grower. Organizations like the Xerxes Society and Pollinator Partnership have programs designed to help growers plant conservation spaces for wild bees. These spaces provide the food, overwintering sites, and nesting habitat the bees need. When bees can find more food, they're stronger, healthier, and they're less likely to become sick. So I've been studying these plantings adjacent to apple orchards for the past couple of years, and the results are looking promising for bees. While this experiment looks at some topics we're not going to discuss today because it's too nitty-gritty and it's kind of boring, I want to highlight how it's relevant and what can be learned from this study in the context of bee health. We'll just go through some of the basic findings for the first year of this study. So in this experiment, we inspect several distinct plots at each of 6 orchards after apple orchards have finished blooming to determine if there's a gradient effect from wildflowers into orchard spaces. Today, I just want to quickly walk you through the differences between the wildflower plantings and their respective orchard spaces. We used to do this, or in the study, we have a lot of different plots, but it's much easier to just look at wildflowers versus orchards directly. So the first thing that we did in this study is we looked at the percentage ground coverage of a few different categories. We looked at this in the plantings themselves and in the orchards. So landscapes get divided into ground without anything blooming or anything growing, excuse me, with just grass, any vegetation that could bloom but isn't in bloom, and flowers that are in bloom. And flowers that were blooming were identified to species. In each plot, we also sample pollinators using blue vein traps. Their pollinators are attracted to the reflected plastic on these traps, and they're filled with a little bit of soapy water. So the bees fly into the blue plastic peaks on top, they fall through a hole, they land in soapy water, and they drown. Bees are brought back to the lab, dried, pinned, and identified. We only do this for 1 day every 2 weeks, so pollinator populations are not negatively impacted because blue vein traps are very effective. They are beacons in these otherwise sea of green places with no food, so bees love them. We get special permission from the state to do this kind of work to ensure that populations are being conserved effectively. It didn't include my permit, but I do have one. Okay, let's look at how landscapes measure up at each of the 6 different orchards we visited. You can see here, as shown by these large, kind of gray-purple patches, that flowers in bloom often make up large percentages of the landscape wildflower in wildflower plantings exclusively. Within orchards themselves, you can see grass was the dominant coverage type, as shown by this blue-green color. The gray-purple color representing flowers is almost non-existent in orchards. And without these wildflower plantings, there just isn't that much available for bees in orchard spaces. Here's another way to look at this information. Instead of showing flowers altogether as a single category, this chart shows each flowering species that was seen at each orchard and for how much of the year they were in bloom. There were more flower species that bloomed for longer periods in wildflower plantings shown on the left than in orchard spaces shown on the right. Now, I recognize that these figures are hard to read at this scale, and that's okay. I just want you to notice that the number and length of the green lines representing blooming flower species was significantly higher in wildflower plantings than in orchard spaces. There are more flowers that bloom for longer periods when flowers are appropriately managed for. So if there are more flowers blooming for longer periods in wildflower spaces, where would you expect to find bees? Well, bees go where flowers are. Bees were significantly more abundant and speciose with longer active periods in wildflower plantings than in orchard spaces. Across all locations, 81% of bees were collected within wildflower plantings, while only 19% were collected in orchards. So what have we learned from the first year of this study? Bees often use flowers which are not intentionally managed for, like clover or dandelions in orchard spaces. However, orchards have significantly less floral diversity than wildflower plantings, and wildflower plantings ensure that orchard spaces remain habitable for bees for the duration of the year. Harnessing the public's altruism towards native pollinators can further facilitate conservation efforts. Public pressure to preserve pollinators has encouraged growers to maintain these wildflower plantings. This improves habitat not just for bees, but other beneficials as well. And I guess I should have mentioned that part of the reason that growers plant these wildflowers is because they receive a certification from the nonprofits like Xerxes and Pollinator Partnership that say we're growing food that is bee safe or bee certified because we plant these flowers that allow them to live even when other things aren't in bloom. So while I've only shown you a small portion of the findings from the first year of this study, we're finding out a lot about the pollinators and other organisms that depend on conservation plantings. The third year of this study just started last week, and I'm hoping to publish our findings next year. I was also hoping to include some of the stuff from last year, but I just couldn't get it cleaned up in time, so you'll have to wait for that one. So future work in this project will also examine the communities of other beneficial organisms which utilize these wildflower plantings, especially those which could be used to control orchard pests. Ladybugs and green lacewings are frequently found in wildflower plantings and orchards and help control pests like woolly apple aphids. They have been commonly found in our traps and our nets, which I didn't talk about the aerial netting, but it's fine. But data analysis on them has not been performed yet. So over the next year, I'll look at all of the data we have on these other beneficial organisms to see if these wildflower plantings aren't just helping bees, but helping the community at large too. This kind of work would allow for IPM or integrated pest management, where wildflowers could be planted to help reduce pesticides even because they would bolster these beneficial populations that are eating the pest organisms of apples. What I really want you to take away from today is that bumblebees are just one of the many groups of pollinators important to our ecosystems and our agricultural systems. Unfortunately, bumblebees are declining, like most bees, due to habitat loss, parasites, and pathogens. However, by increasing habitat for them, you can bolster their populations and keep parasites and pathogens reduced. By participating in groups like this one, the Washington Native Bee Society, you're already doing so much more for bees by learning about them and working to improve their lives. Conservation plantings are becoming normalized as consumer interest in them grows, and they work. It's not only up to one individual person to save bees, it's up to all of us. If you're curious about the topics discussed today, more information can be found in the publication that I mentioned on WCU's Extension website. You can also contact myself, the Xerxes Society, or Pollinator Partnership if you're interested in habitat augmentation programs. And again, I'm sorry, my email is wrong, but it's got a period in the middle. So if you want to contact me, it's just mario.luppino@WSU. But I'll take any questions you guys have now.
Excellent. Thank you, Mari.